Good evening, students.
Good evening, sir.
Good evening. Right, yes, let's begin with the class today. And sorry, I had a issue in connecting to internet because I'm happen to be at my home in my village. Sometimes I have difficulty in having the access to internet. Okay, and especially the course page and the Zoom page don't open. Although I can have access to some of the other website, looks like it's the demand for the internet speeds to me. It happened once before, and today fortunately don't have that problem and able to access the internet without issues since the last few hours. So yes, and second one I wanted to tell you, haven't actually still finished our papers. I'm almost done through them, so we should be able to discuss them in tomorrow's class. So wait till tomorrow. So I'm going to send you the answer scripts to you, so have a look at them. So far I think most of you are writing well. Few changes are required. Some of you are writing really well, and I'm mentioning that what mistakes you're doing in each of those specific questions. So I'm going to create a folder and post those answer scripts together. So you can have a look at how others have performed as well, and so you can learn from each other. Okay, right. And also, given that I missed a class, I'm actually thinking of postponing your exam by a day or two. Let me see. Is there any other exam that is scheduled this week for you? Okay. So is it okay if I push the exam by, let's see, Thursday we're thinking of, right? Either Friday or Saturday, most likely Saturday. Okay, so that we can have continuously classes for the rest of the week and then have a exam test posted by 1 or 2 days. If that's okay with you guys, okay? Right. So that before that we can finish discussing your scripts and take few more classes and then have the test without disturbing the class. Okay, right.
We
So
have any test, Vishal, Shweta, do we have any test this Sunday?
No, we don't have.
Can we have it on Sunday, sir?
yeah, that will be better. Yes. So we can have the test on Sunday and finish our classes as usual for the rest of the week. Let's do that. Okay, like last time what we have done in the morning, I'll post the test. So you finish writing it on Sunday. Okay, that will be better. Great. Okay, where did we stop the last class? I think we were discussing the ABO blood group system, right? So we started with the genetic markers. So we finished talking about DNA fingerprinting in the last class. We started with the genetic markers. Do you remember what are genetic markers? If you have to tell me in one sentence, what is the genetic marker?
Regions with great variability.
Okay, regions with great variability. Okay, that is one way of answering it. Yes, but how is that variability useful to us? So I think you are mostly, if I'm not wrong, you're talking about the DNA fingerprinting principle, am I right? Where there's more variation that can be identified or probed for DNA fingerprinting. Am I right?
Right,
So
right.
marker can also have variability, but marker is a reference point, is a point or the physical place or location that can be used as a reference for identifying maybe a gene involved in a genetic disease. Okay, so marker is basically a reference point to us. So what are good markers, and how do they vary from population, or how do they vary in distribution? What is the use of them? Okay, try to understand these things. And as I was telling you, fortunately they're not asking too many specific questions on this from this unit. Otherwise this unit can be a bit challenging. Okay, but to be safe, I don't want to leave out anything. So to be safe, in case if they happen to ask anything on this, we are going to briefly go through them. So what are we learning? I think ABO blood groups and RH blood groups in a way easier for you because you have some idea on them. In the last class, we were discussing about ABO blood groups, categories and types. Let me share the presentation with you and see where did we exactly stop the class. All right, so we discussed about the basic idea about antigens, right? Presence of antigens. If A specific antigens are present, A blood group, and B specific, like that, both A, B, O we talked about. So antibodies will always be opposite, meaning if person with A blood group will have only antibodies for B, correct? Like that. And remember the universal donor should have all three antigens or no antigens. Donor should not introduce any antigens into the recipient, otherwise recipient's immune system will react to it. So all three negatives, A negative, B negative, O negative. In a way, negative for all three. So that is O negative. Opposite is true for the recipient. Recipient body should be familiar with all the antigens. So no antibody should be made. That is AB positive is a universal accept. Correct? But because of the rarity of some of the antigens, some of the blood groups, maybe O positive is more commonly used than the O negative as a donor. Okay, we also talked about a blood group. Do you remember the blood group where even O blood group person cannot donate blood to them? What is that blood group that we discussed?
Bombay.
Bombay blood group.
Bombay blood group, correct, exactly, Bombay blood group. How is that different from the any of these blood groups? Do they have A, B antigens on them, the Bombay blood group people?
They don't have H antigen, I suppose.
Okay, so it is something to do with the H antigen. So Bombay blood group people, do they have H antigen? Okay, people with O blood group have H antigen, so they're capital H. That is how we say. Bombay blood group people usually have small h. They don't even have this component that is seen to be present in O blood group. So in a way, although we normally discuss that O has no antigen, but it has H antigen. Okay, and AB is more complex than H. It's like adding extra components to the antigen for the A blood group people and antigen for the B blood group people on top of H. But even this component is not present in Bombay blood group people. They are missing this brown region or red region that you are seeing here. So for them, even O can be seen as an antigen. Okay, so it's a rare blood group that is usually found in Parsis in Mumbai. Okay, right. So, okay. So let's focus on the polymorphism part on the blood groups. So here I have picked up some information on how ABO blood groups are distributed around the world and what about in India. Okay, so we'll get a brief idea on distribution of ABO blood groups and in the world as well as in India. Let's have a look at it. So what are you seeing here? You can actually notice that. So we are seeing the distribution of these. Here we're talking about first A blood group. Look at the color code. Okay, look at the color code. If it is slightly grayish or yellowish, so this is the lightest percent or lowest percentage we are talking about, anywhere between 0 to 10%. You're talking about bit of slightly reddish or brick color. If you are noticing it here, so we are talking about intermediate colors, and more darker colors that you notice here is percentage that is above 30%. So A frequencies is more common if you see in the Australia, part of Australia that you notice here, and part of North American regions that you see here, are the regions that you see in the European regions, are the regions here that you notice. So this is where highest amounts of A frequencies are seen. So European population, some European populations, you see some dotted structure within the Europe. So some European population, some North American, especially North American Indians, Okay, tend to have higher amount of this, and Inuit populations in Arctic North Americas also seem to have higher amount of A frequencies, and Australian Aboriginal tribes and Australian Aboriginal types. Okay, write down few examples like this in polymorphism. So high frequency of A blood group is seen in, so most importantly, you can write some North American Indian groups, Australian Aboriginals, and some European and Inuit populations. They tend to show highest amount of A blood group. Interestingly, you will notice that most of the South America has a very low amount of A frequency or A blood group. Very low. You will notice that what is more common in South America. South America shows the lowest amount of A blood group population. Whereas majority of the other areas we have more or less moderate to high levels of A. Here, India is also partly in the middle. Okay, A is also less common here, otherwise Central Asia. Okay, so A frequency seems to be more common in these regions. And then what about B blood group? Where is it more common? Can I move on? We written this down briefly. Okay, what about B and O? Let's have a look at them. So high frequency of B, where is it seen? Observe the image here. Briefly look at the image. So lighter colors, more or less B is not much seen at all in the Americas, as you see. Mostly, I think you see in the Asia, Central Asian region, partly in Africa, you see B, correct? So high amount of B is mostly here in Central Asia part, 20 to 25%. Few pockets of it here towards, I think, northern part of India and above that, especially in the Himalayan region, that you need is higher amounts of B blood group. Okay, and a few in the sub-Saharan African region. You will see few of these pockets of B, but Central Asia. So most common that we are seeing high amounts of B blood group people living in the Central Asia part. But rest of the regions, mostly in Americas, you don't see it at all. So A is distributed here and there, not much B in Americas as you notice. And in Asia, we have mixtures of things, bit of A, bit of B. Okay, what about O blood group? So O blood group is common actually, distributed to many areas. Okay, and interestingly, you see it here, the one that's standing out is South America. It's very common in South of the highest, the most of the South Americans seems to have, they're monomorphic to O blood. Okay, otherwise part of southern part of India, central to south in India is also more incidence of O blood group people. Otherwise, O is definitely more common because O is given that it is it's an allele that can be present in along with A and B. So O can be seen even with people with A blood group and B blood group, right? A, O, B, O genotypes can still exist. So O alleles definitely are more common, but also O blood group. And what is standing out here is the Central America, sorry, South America and part of the Central America, but mostly South America. 90 to 100% of them seems to have O blood group. Okay. So this could be something like because of founder's effect. The people who traveled and who probably have the O blood group tendency to marry within them may lead to this type of allelic distribution. Okay. So I was looking into if there are any studies which analyze the blood group distribution in India. We kind of got an idea about this, right? Blood group distribution here. I think we have higher amount of O blood group in the southern part of India, and that we have seen in the past in the high frequency of B blood groups are seen more towards the northern part of India that we have seen, right? Yeah. Okay. So I will show you another study. Okay. This is just for an idea. Don't write anything from here because I felt a more newer study had come, but I just wanted to point out at the overall distribution of the blood groups. Seems like O is the most common blood group, about 60%. These numbers may vary depending on the studies. And this I'm talking about 1990 study, 88 study. Okay, this might have slightly changed, but still what you notice is we are people with more of O blood group and then A blood group, then B blood group, approximately, roughly, we get this idea. So 60% of the share goes to O blood group and about 20 or below 20 for the B and about 20 for the A blood. Okay, that's how I think the distribution is, but this is a bit an old study, couple of decades old study. I was looking for something new, and I have noticed that, I think before moving to that, you can also clearly see that here. So the big chunk, O blood group or B blood group will check the big chunk in India. This is for Indians. Okay, and Australian Aborigines, mostly O, Native Americans, mostly O, part of Africa O, and Southeast Asia also O, Laos especially. Okay, otherwise English where you see more of A blood group. So you notice that there is some peculiarity with respect to the blood group presence in a population. How that is useful to us in anthropology? I leave you with the question given the distribution. We'll come back to this idea. So this is one study that I found published in 2014. Study published in 2014 by Agrawal and et al. So as per this study, they have done an analysis on Indian population on all four regions, northern part, southern part, east, west, and also in the central part of the country. So 5 zones that they looked into. So what did they notice here? They noticed that O is the most prevalent group in all the regions of the country. So they didn't use color code here, but if you see the one on the rightmost in the AB blood group, then O, then AB, sorry, then B, then A. So AB, O, B, and A. So if you notice the O, the percentage for the O is actually more or less south. We see more of this, and also in the eastern part, western part, and also the central part. So O definitely seems to be more prevalent. And then later the B. B is the one that probably shows recent presence in India. So O is the most prevalent group in all regions of the country, and except North, where B was the more predominant group. The northern part of India, where B is the more common. Okay, so what do? They don't actually analyze entire population. This is a sample-based population. Sometimes it may be a bit misleading, but we work with the data that we have with us. Correct? So what else is important? Of course, we are only focusing on ABO, but if you see in Indian scenario, RHD, are we more RH positive or less RH positive? So RHD negative population in the country is very, very small. About 5% of Indian population or up to 7% are RH negative. Otherwise, up to 95% of us are RH positive. So we'll see more and more RH positive people. Maybe most likely O positive, and then B positive, then comes probably the A positive in India. Compared to the negatives, only probably 5% of the people have RH negative blood groups. Okay, so this seems to be the Indian scenario roughly. So now we went through kind of statistics and distribution of blood groups around the world and in India. Okay, so let's see. Let's look at few reasons why do we have such an amount of variation? Is there anything known about it? Okay, let's see. So Native Americans, we talked about that they have type O blood group. I was already telling you this could be because of founder effect. Right? Small population, small size population might have migrated, and then intermixing within their population might have kept this blood group going. Okay, so reflecting the genetic makeup of the relatively small population that likely settled in America from northern Asia or other regions from where they migrate. So this could be one explanation to this is the founder's effect. Small population establishing that, and then eventually leading to more and more intermixing. But this may not be the case for future reasons. We have to see how this is going to shift. Because keeping O is actually very difficult, because even small mutation or mutation in O to become A can result in A blood group and also B blood. And any other observation that we have noticed, people where there is a balance between A, B, so more common in these populations are not O, but AB blood groups. There it could be something to do with the bacterial disease. So this is a slight correlation that is given here. I totally suspect if we come to conclusion on this. But a balance between A and B blood groups are more common where there is more incidence of bacterial infections. If a population is experiencing more bacterial infection, seems to have more of A and B. Somehow A and B blood groups seems to provide some resistance to them. Okay, these are all simple ideas. And O seems to be more common in populations with more viral diseases. Some loose correlation that we can make here. Okay, so AB seems to be more common, have correlation with more bacterial incidence, bacterial disease incidence, whereas O is more common where viral diseases are predominant. Like that. But I think nowadays, I don't think we can categorize areas based on viral or bacterial disease. Few regions, Ebola, Zika, so these diseases more predominant in some areas than the other areas. Okay, but I don't think they are still very concrete. Okay, definitely there seem to be some preference towards the disease burden rate and the type of blood group. There may be some sort of natural pressure acting on these populations. That likely, explanation to this. Okay, so and we have seen that. I would like to ask you the significance, use of knowing this. Why do we need to know? And if you, in case I've seen that they ask questions on ABO blood groups being, why are they good markers? Why can we, or what sense they are very good sources for information to us? So look at the diversity of ABO blood groups. Populations, different populations in the world seem to have some sort of trend with respect to what type of blood groups they have. So they help us to identify the region from where they might have come, right? It should give us a bit of an idea on that. Second, this ABO blood group diversity would also help us solving some issues. Okay. First, let's look into the paternity issues. How can we deal with the paternity things? Okay, so I'm going to give you some pointers for you to write here. Can blood groups be good markers for solving paternity issues? What do you think? Is a blood group is a good source for solving paternity issues? Can you identify if a person is biological father of a child? Is it possible with blood groups? We talked about this scenario with the DNA fingerprinting. What about the blood groups? This topic is from, as I was telling you, from Unit 12. Paternity diagnosis. So what is the role of blood groups in paternity diagnosis is my question. Can we use blood groups to prove paternity, to show if someone is a biological father or not? Is it enough? Let's see with examples. So let's say two parents, okay, father and mother both are blood group O. I'm going to explain a couple of examples here to tell you how blood groups can be used in solving the paternity issues. Understand them, how far they can help us, and what are their limitations. O blood groups. So father and mother both are O blood groups. So in this particular case, father in question, let's say. So possible children. Can children have any other blood group than O blood group if parents both of them have O blood group?
One of the allele might be O and one might be A or B in parent.
Sorry, I didn't get that. So if parents are O blood group people, can they have children without O blood group? Can they have A blood group child and B blood group child? Both father and mother, if they happen to be O blood group people.
No, sir.
No, no. Okay. Correct. Because O is the, remember how does O express? O is the homozygous recessive, only in the homozygous recessive condition. Two O alleles are needed to express it. So if father is O and mother is O, there is no way they can have A allele or B allele. It means they're homozygous for O. So genotype of the father will be OO and genotype of the mother will be OO. So there is no way they can have a child with A or B. They must be O blood group children. Okay, so impossible to have AB, AB. So we know if both parents are O blood group people, the child is O, in no way they can have a father like that. Okay, but yes, there's some twist to this anyway. So I'll tell you that in another examples. So person with O blood group and A, one parent is of O blood group, one other parent is of A blood group. Okay, they can have both O blood group children and A blood group children. Try to write down yourself, okay? Brief analysis you write down as I'm explaining it to you, so it will become clear. So write down the genotypes when I'm saying it. O blood group mother can have only one possible genotype, OO. Let's say mother is O blood group, OO must be her genotype. What about A blood group father, let's say? What are the possible genotypes for A blood group? Possible genotypes for A blood group.
IA, IO, and IA, IA.
IA, IA, and how is the second one?
IA, IO.
IA and IO. Okay, right. So I is normally what we use here, but if I simply use the word A and O, both A homozygous for A are heterozygous. One A, one O alleles. Because A is the dominant over the O, so what happens? So they can also have A blood group. So meaning father can have one O allele with him, right? So that is where if O allele meets with the O allele of the mother, then child will be O. So they can have a child with O blood group, or if A joins with the O allele of the mother, then it will be A blood group. So O and A blood group children are possible. This is straightforward. Parents are OA, children can also be OA. There shouldn't be no confusion here. But let's pick up something that is more complicated. So OB is also same story. Parents, if they're O and B type, children can be same. What about O and AB children? O and AB father, parents, sorry. So mother is O blood group, father is AB blood group. Can they have, so can they have children with AB blood group? If father is AB, mother is O, or vice versa, doesn't matter. One of the parent is O, one of the parent is AB. Can they have AB blood group children? Don't look into the table possible children. You think about it because I want you to answer this. And with reason. So is it possible to have AB children, AB blood group children to the born to the O blood group parent and one O blood group parent, one AB blood group parent? Yes or no? Try it out. We're just trying, okay, so that I can explain you if something goes wrong. Sunil?
No idea.
No idea. Okay, that's good. Okay, anyone else who can answer? I'll explain that to you, okay?
But you said mother is AB and father is?
Oh, can they have children with AB blood group? So one parent, you write down this. Okay, O crossed with AB. O crossed with AB. So write down the genotypes. Only one possible genotype for the O blood group parent. What is that? Homozygous recessive. So
So
2O. OO.
if we take AB, then we would be having two alleles, A and B.
Hmm.
And if we cross OA, then A would dominate over O.
Hmm.
So so AB cannot be possible. Either A would be possible or B would be possible.
Exactly. If you do the cross like that we learned in the Mendelian genetics part, you should be able to answer this. So you'll have answered before you. Sunil, I want you to try this. If you figured it out. Okay, we are just using the problems that we solved from the Mendelian genetics part. Are you following what I've said so far, Sunil? We take two O's. O blood group should be only homozygous recessive condition, two O's, and AB should be 1A, 1B allele. So genotype should be easy here. You write down the alleles that are possible. From the O blood group parent, only one O, one allele is possible, that is O. From the another parent, two alleles are possible. A allele, a gamete with A allele, a gamete with B allele. So you cross one of these, right? Either you take A or B. On the other side, the first parent had only O type of allele. So A has to meet with O, resulting in AO gives rise to A blood group. B, when you take the B gamete containing B allele, the outcome should be B. So A blood group is possible, B blood group is possible. But we have no possibility for getting AB because both parents should have A and B alleles to give rise to AB combination because each parent contributes one allele, correct? So in this case, do you agree, Sunil, that A, B child is not possible?
Right.
Because only one parent is having both A and B alleles. They don't mix again. So what are they mixing with? They're mixing with O allele. So AO, BO, possible. These are the possible genotypes. So AB blood groups are possible, not AB together. A or B is possible. Is that clear to all of you? In the exam, when you are asked to write about ABO blood groups and their importance, explain this with an example like this. Okay, I'll give you an opposite scenario to this. Okay, see if you can figure it out. So B blood group mother, AB blood group father. What are the possible blood groups? B blood group mother, AB blood group father. Write down the genotypes first. B blood group, what genotypes are possible? And write down the heterozygous condition. Because for B, both possible genotypes are BO and BB. So take the BO, not the BB, because then you will get all the possible outcomes. So take BO for the B blood group mother, and AB, of course, you only have one choice, AB.
So possible children can have AB blood group, A and B,
Very good.
3 types.
Okay, what about others? You get that answer? And don't worry, if you didn't understand it or if you didn't get the right answer, let me know. I'll tell you where you have, where you went wrong. Okay, so what are the possibilities?
AB and B.
A or B or AB. Okay, all three possible. We don't get O blood group children. Is it possible to have O blood group child? Although there is O allele possible. Mother with B blood group can have BO, but father is not contributing any O allele. We need two O alleles to show the O blood group, right? That is not possible. O allele is there, but not with both the parents. So O blood group is not possible. So what are we learning here? We are learning that based on the blood groups, you can figure out the possibilities. Like we just talked about that B blood group mother, AB blood group father, or vice versa, cannot have a O blood group child. So similarly, we talked about in the case of, let's say, AB blood group scenario that we talked about, right? So if father is, one of the parent is A blood group, another is AB, okay? So again, O blood group is not possible. And both the parents are BB blood group parents, AB is not possible. Like that, there are certain blood groups and certain outcome is possible. So meaning you can calculate that. You don't need too much of a knowledge for this. Just the amount of knowledge we are discussing in the class today is enough to figure out if a so-and-so person can be biological father or not. And most of the times, I remember I was teaching this in one of my science and technology classes, and one student came. She was unsure about her blood group because they make a mistake with respect to the writing genotype. Because heterozygous, if you write, so she said, my father and mother both have A and B blood group. I'm O blood group child. How is that possible? Is it possible? If father is A blood group, mother is B blood group, can they have O blood group child? AB blood group people can have all possible combinations because they are like this AO and BO. You consider AO genotype, BO genotype. So two O's can meet, one from each parent, giving rise to O blood group. So that is very much possible. But they don't know the genotype concept here. That is why they get confused. Okay. Otherwise, anyway, B can meet with AB. So AB is very much possible. Two O's are there. So B can go with O, A can go with O. So two O's, two A are AO, BO, AB. All of these genotypes are possible. This is the only combination where you will see all of them. Okay, right. So what are we understanding here? Blood groups can help us in solving paternity cases, but with the limitation. So can they really prove? Can they really prove it? They cannot prove. But what do they? They can give us clues. We can only rule out. It can provide evidence if a male is not the father of a particular child. So if this blood group father, is it possible to have a father if child and mother have these blood groups? We can have that. Let's say if the, in this particular case, which one of them? So we'll take the child is O blood group. Child is O blood group. Mother is B blood group. And the person in question, the father is of AB blood group person. Is it possible to be a father, biological father? Yes, very much possible. AB blood group father with A blood group mother can have O blood group child. Sorry, cannot have O blood group child. Correct. So AB is not a possibility. AB combination is not a possibility. What do? You say he cannot be biological father. No further testing is needed. So blood groups can eliminate that. Blood group can say whether he is likely potential or not, potential to be a father or not with that given child. They can rule out the possibility here. It will not prove. ABO blood groups cannot prove the paternity issue, but they can only eliminate the wrong options. So actually they can tell us whether we need to go for further DNA testing or not. If they say that ABO blood group, this person cannot be the biological father based on the blood groups, you can rule out the test need for performing DNA fingerprinting test. So they can only provide as an evidence to check, and a male is not the father. If he's not a father, cannot be the father because he is a O blood group child, mother is A, and AB cannot give rise to a child like that. So you can say that this person cannot be the biological father. You don't even need the DNA fingerprinting test for like that. In the negative cases where he is not the biological father, blood groups can help us rule out the possibility. But they cannot prove, they cannot tell who is the biological father. We definitely need DNA fingerprinting. Or other markers. I'll tell you, we're going to learn about a few other markers. The child cannot have a blood group antigen that is not present in one or both the parents. I was telling you this, if both of the antigens should come from the parents. Okay, but heterozygous condition usually confuses a bit. A naught, B naught here can have all of them. So in either in heterozygous condition also, one of the parents or both the parents should have this antigen. Children cannot have antigen that is not present in the parents. So next, proper identification of blood group is key. So all this depends on whether they properly identify the blood group or not. If not, they would confuse us further. It is easy actually, indeed it is very easy to identify the blood group. They use antibodies. They add antibodies to the antigens. Quickly, in a couple of minutes, you can find out your own blood group. Okay, right. So along with the blood antigens, A, B, O antigens, other markers like HLA markers that we briefly talked about, proteins, other proteins, plasma proteins also used in the blood markers. So together with them, we can identify the biological father with more certainty. By itself, blood groups cannot solve it, except if we can rule out if a person can be biological father or not. Otherwise, along with other blood group, other genetic markers, or DNA fingerprinting is ultimate here, can be used in identifying the biological father. Okay. Right. So that is a great benefit of ABO blood groups, except we have some limitations. They cannot provide ultimate answers to the paternity issues. Okay. Right. So a couple of things that we talked about here. One, there's enough polymorphism with respect to ABO blood groups, and they're very, very important in a couple of ways. One, we already discussed about it, blood transfusion, compatibility of with the parents. Okay, sorry, compatibility between the donor and recipient, it is very important, and is also important in studying the population. Maybe there is some correlation with the infection, disease infection rate in a given population, and they also are useful in solving some paternity issues as well. Like that, ABO blood groups are considered to be very good genetic markers because they're very commonly distributed. Of course, there are exceptions to this, but it's commonly distributed, showing enough diversity and have good applications. Okay, so for these reasons, you can consider ABO blood groups are good markers, good genetic markers. Okay, when question comes, highlight on these points and take the examples of things that we talked about in this session. Okay. So the other part of the ABO blood groups we need to talk about is the RH blood groups. Right, so either it is RH factor positive or RH factor negative. So far, I think in the classes before we talked about RH factor gives either positive or a negative blood group. Right? In the last class we discussed about RH positive or RH negative. We talked about. Correct? But what gives rise to the RH factor? How many alleles are present? How polymorphic it is? Let's have a look at it. I think we kind of talked about it. So O is more common. RH positive is more common over the RH negative. Correct? We talked about that. So what results in RH blood group? Let me give you a brief introduction to that. So RS factor is considered to be the clinically the most important blood group allele of blood group system. Even compared to the ABO blood group, clinically RH factor plays an important role because sometimes looks like, I don't know whether I talked about erythroblastosis fetalis. I think I remember talking about this in the one of the classes where we talked about the incompatibility. Maybe in the genetic load class, I think I've talked to you. Correct? Do you remember
Yes.
about RH incompatibility problem? A mother who is RH negative and the child who is RH positive can have some cross reactivity of antibody to antigens. Okay, compatibility issues may be seen. This is compatibility issues can be found between any other blood groups indeed, but RH seems to be more risky and troubling. Okay, so clinically, RH factor is the most important of the all ABO blood systems, and this is also the largest of. There are 29 blood group systems. They say they talk about, and largest of all of them seems to be RH. Lot of diversity that you notice there. So there are about 49 antigens. It is very complex. I'm going to give you a brief picture about it. We don't have anything in the literature. Also, they don't talk much about the total complexity, but a generic idea is provided. I'm going to talk to you about that. So this is definitely have several antigens. Lot of diversity can be seen here. The outcome is seen as very simple: RH positive, RH negative, but allelic combination seems to vary so much. How is that possible? Okay, so many different alleles, but only two outcomes, positive or negative. How is that possible? Let's see what scientists have talked about. Okay, so this Rh antigen or D antigen was discovered later in 1930s, like basically in 1900, 1901 or 1900 when blood groups were discovered. But 1939 they discovered the Rh positive and Rh negative. So Rh factor was discovered in the 1939. Karl Landsteiner, if you remember, Karl Landsteiner is a common uh, question for the competitive exams. Who discovered blood groups? Karl Landsteiner. You might have heard about it. I don't think this factual data is needed here, just mentioning it. Landsteiner, Landsteiner. Landsteiner. Okay, not needed. You don't have to write down. Okay, but they discovered it in 39. So let's see what defines it. What did he find out? So he was saying the D antigen is an important antigen, but there are many antigens. They are talking about not just D, C, E, D. Three types of antigens are present. RHD, RHCE are present. I'll explain you briefly on RHDE and CE concept. So totally there are three types of antigens, C, D, E antigens. C, D, E, main antigens, otherwise there are many nominal alleles are present. C, D, E seems to be the most important ones. And out of them, D is a key antigen to define the outcome. So if D is present, capital D is present, it's Rh positive. If capital D is absent, it's Rh negative. Doesn't matter what other alleles are present. I'll show you the combinations of alleles. Okay. So look at this. This is what given by some scientists, Fisher and Race or Wiener has given. Let's look into the Fisher and Race concept here. So look at all these possible combinations. Rh positive combinations, Rh negative combinations. Rh positive combinations, look at this. So what is common to all these four possibilities? Capital D. Doesn't matter what is the other allele, whether you have capital C small c or capital E small e, doesn't matter. So C, D, E, look at this combination. Rh positive. All three capitals, only one capital is sufficient. Doesn't matter what is the rest. So Rh positive means it's defined by presence of capital D antigen. Excuse me. Okay. So rest of them, as far as there is no D, rest of the possibilities are gives rise to Rh negative. So write down these 8 combinations, all small, both C and E capital. But as far as there is small d, all of these combinations will result in Rh negative. So this is a bit simpler idea about it. I think the actual picture seems to be more complex for Rh factors. Okay. Right. So this is briefly about the Rh factor genotypes. What gives rise to Rh positive? What gives rise to Rh negative? Okay. Right. But altogether, they say that it is also very highly polymorphic. 170 alleles have been found in RHD. So for D itself, RHD, there are 170 alleles. So what we have seen is a very simple picture of it. Okay, several alleles are present for one location here. So different people may have differences in them, but the outcome is normally seen as Rh positive or Rh negative. Okay, I was also looking for any particular statistical data about the distribution of Rh factors in different populations. I found that. So most common, we talked about Asians, including Indians, and even Africans have highest amount of RHD, Rh factor positive is very common. Native Americans also this is common. African Americans also show about 90% of Rh positivity. Okay, even Europeans slightly less, and people in European region, these people living in Spain, Basque people of Spain and France have these are the people with the highest amount of Rh negative blood. So they have 35% of the Rh negative blood, about 35%. So this is a population where you don't see Rh factor too much or not as significant as in the other places in the world. Okay, otherwise most of us actually show Rh positive is more common. So why do they have actually, why only these people have about 30-35% of Rh negative blood group? Maybe it started there. We don't know. It may hint that it might have started there. So still, I think Rh factor, even if you have one positive Rh factor parent, so that is sufficient to have more children with the Rh factor. Correct? Presence of factor or one antigen is sufficient. If it goes to the next child, even though Rh factor positive plus Rh factor negative parents are married, so the higher percentage of having Rh factor children. But of course, it depends on the heterozygosity and homozygosity condition as well. But definitely there is higher possibility. Okay, maybe Rh antigen might have originated here, but otherwise it's more common to see Rh positive people. Anything known with respect to the distribution, with respect to the polymorphism related to them? I've collected a few points on this. So Rh system, Northern and Central European populations, in a way they differ from each other with respect to what alleles they carry. What alleles they carry. Most Northern and Central Europeans, they differ from each other only slightly. They contain CDE, small c, small d, small e. So Rh negative or Rh positive? Rh negative, right? No capital D. So about 40% of them, 40%. Okay. So whereas if CDE 40% common, is the rest of them probably got distributed among the other possible combinations. Africans tend to show C capital D capital E. So meaning Rh factor positive. Two small c and e's and capital D seems to be more common in Africa. African seems to have C, D, E, C, small c, capital D and small e. Whereas the CDE, that which is 40% in the European population, is only 20% in the African population. Only about 20% in the African population. Whereas in Asia, especially Eastern Asia, CDE, small CDE, which is 40% common that we talked about in the European populations, like some of the populations I mentioned to you. Okay, so CDE is more or less absent in the Asian part, especially Eastern Asia. So if CD is more or less absent, is erythroblastosis fetalis, that incompatibility issue between Rh factors. Will that be more common in this population? No, it won't be, because more or less we'll have people with Rh factor positive. So very less chance of having Rh negative mother and Rh positive father. So the erythroblastosis fetalis condition seems to be less common in eastern parts of Asia. Okay. Right. So this is briefly about the rhesus factor. What is Rh factor? And what type of genotypes or what type of allelic combinations we are seeing. So basically what is polymorphism with respect to Rh factor, and specific information with few examples of how this is distributed among various populations. Okay. And what we are seeing, this is clinically very important because erythroblastosis fetalis happens between Rh positive father, Rh negative mother when they happen to have Rh positive child. This is important. Some population-wide variation can be used in studying them. Okay, so this is something that I wanted to tell you here with Rh factor. Next, we'll move on to the next marker, that is MHC, major histocompatibility complex, that we kind of briefly talked about. Right, MHC we briefly talked about few days ago in the immunogenetics part, right? In the immunogenetics part, we discussed about the MHC. So I'm sure you probably have remembered about MHC. So short note is possible on MHC. So what would you write for MHC? So major histocompatibility complex. Let me briefly explain that and give you more points than what we discussed in the about it in the previous classes. So in all vertebrates seems to have this region, a genetic region where there are some genes that code for some proteins which are responsible for deciding whether a graft can be accepted or not. Meaning if you have taken someone's organ, you received someone's organ, that is called graft. Is that graft accepted by others? Is it acceptable or not? Okay. So this region which defines the acceptability of the graft is called major histocompatibility complex. I was telling you the meaning of it, right? Histo means tissues, compatibility complex. So this marker or this antigen is responsible for defining whether a given tissue is compatible or not. That is what's called histo, tissue compatibility complex, meaning it's not simple a few molecules, it's a combinations of molecules. It's a complex. It's not a single molecule. Unlike A antigen, B antigen we talked about here, it's more complex, much more. This is probably the most complex of all. Histocompatibility complex and the major one, major histocompatibility complex. There seems to be a lot of varieties in them, so this is considered the major one. So people who have better match in their tissues with respect to MHC. So between two people, if MHC matches better, there's a higher chance of success in receiving the organs. That is a key use of MHC. Along with that, we kind of talked about its role in immune system functioning. I'm also going to explain you a bit in detail here. So people who are identical or who have more matching in MHC, in case of MHC, there can be more success in donating organs within them or receiving organs. Okay. In addition to this, in addition to the survival of the organs or grafts, MHC also plays an important role in antigen recognition by our immune system cells. So our immune system to recognize specific type of tissue, whether it belongs to or not. I was telling you the self and non-self discrimination can also be done with the help of MHC. So our immune system can recognize whether this is my MHC or not. So do I have to attack it or should I have to spare it? So that immune system functioning seems to be also controlled by or defined by MHC. So let's learn a little bit more about MHC, the types of categories of MHC. Okay, this part I've kind of already discussed with you in the class where I talked about immunogenetics part, right? Let's get into more details about MHC. So there are two classes of MHC. There are two key classes of MHC we call functionally. We're talking about function of MHC in the immune system. Okay, two classes: class 1 MHC, class 2 MHC. We talked about. As you see in the box here, class 1 MHC, class 2 MHC. Two types of classes are important for immune system function. So where are these class 1 and class 2 MHCs are found? Class 1 is found in almost all cells of our body, all nucleus containing cells. So there are few cells which don't contain nucleus like RBC. Okay, otherwise all nucleated cells contain class 1. So class 1 is more common, more commonly found on our body's tissues. Whereas class 2 is special. Class 2 is found only on APCs, or antigen presenting cells, we call them. Where are class 2 MHC molecules found? They are most commonly found on okay. So what do? They have different functions, the way they react to the with the immune system components. So MHC1, class 1 MHC molecule, okay, they are present on TC. There are two types of T lymphocytes. Remember, we were talking about in the immunogenetics part how B cells make antibodies, and there are two categories of T cells, T lymphocytes: T helper cells, T cytotoxic cells. You remember them? The mainly two categories of T cells: T cytotoxic, T helper. T helper, as the name suggests, T helper is to activate both B as well as the both B cell mediated and T cell mediated immunity. But those T helper cells carry class 2 MHC. So T helper cells carry class 2 MHC. They're called CD4 plus cells. They have another protein molecule called, or antigen called CD4. They call CD4 positive. The T helper cells. So T helper cells carry class 2 MHC molecule, whereas T cytotoxic cells carry class 1. So their function is different. So always helper T cells' job is to activate proper immune system channel. So for them, they use class 2 MHC. Whereas cytotoxic T cells are specifically designed to act on viral infected cells, cancerous cells, they use class 1 MHC. So they can differentiate whether they're dealing with our own cells or different cells. Based on that, they'll attack. Okay, that is what I was trying to show you in the image. So T helper cell always reacts with the molecule containing class 2 MHC. T helper cell utilizes class 2 MHC. Whereas T cytotoxic cells using class 1 MHC. Okay, so they're important in defining the proper immune system responses, not only graft rejection, graft acceptance. They're also very key in protecting our own cells being attacked by our immune system, and also to identify the foreign cells as foreign. They look for, is it my MHC or foreign MHC? Like that, MHC would define our immune system activity against the foreign cells. They also protect our own cells being attacked by our immune system. So they play a very important role in this immune system functioning, and also in the graft rejection and graft acceptance. Okay, so this is briefly about its role in the immune system function. Now let's see how diverse are these MHC molecules. What type of polymorphism can be found? Okay. So let's look into the genetic basis of this. So HLA gene seems to be present on chromosome 6. So the genes that encode various HLA components are MHC. Remember, HLA is another name to MHC, human leukocyte antigen. HLA stands for human leukocyte antigen or human WBC antigen. Leukocytes are WBCs, right? So human leukocyte antigens are WBC antigens. They are called HLA. Is another name to MHC. So these genes seems to be present on chromosome 6. Chromosome 6. Okay. I'll show you how diverse are they. So just one point I wanted to make a note here. Remaining we're going to discuss. HLA, these genes are present on chromosome 6. How diverse are these? So very, very diverse. Actually, indeed, most diverse. Okay, of all that we are studying here, this is the most diverse one. Where do they get so much of diversity? They're both polygenic and polymorphic. So polygenic, multi-allelic. So they're combination of polygenic, meaning multiple genes code for these proteins, plus there are multiple alleles for each gene. So multiple genes plus multiple alleles together. Okay, excuse me. So polygenic plus multi-allelic. That further increases the diversity of it. Okay, so we talked about MHC class 1, MHC class 2, right? Class 1 defined by 3 categories of genes, A, B, C genes, HLA-A, HLA-B, HLA-C. So MHC class 1 molecules are defined by HLA-A type, B type, and C type genes. Class 1 defined by A, B, C types of HLA, whereas class 2 is defined by HLA DP, DQ, DR. So the D locus, it is D locus, but DP, DQ, DR, PQR basically. So instead of labeling A, B, C, sorry, like A, B, C, they have labeled D, but within D, further combinations, DP, DQ, DR. Okay, so let's get the further information on this. So we have class 1, class 2 like that. So let's look at how many alleles. This is only to present you the diversity. Okay, I don't think you can remember or by heart these numbers. Just to show you how diverse this is. So I was talking to you 1 and 2, at least 2. Scientists have found the third locus as well. But otherwise 1, 2 are most important. Locus 1, locus 2 are more important. And all are present on chromosome 6, as I was telling you. Chromosome 6, 1, 2, 3. So A, B, C, you see here A, B, C, their order is different on chromosome 6, A, C, B, they look into. There are the ABCs and DP, DQ, DR. And look at them, within them multiple genes, alpha, beta for DP, alpha, beta for this, alpha, alpha for this. Okay. So this, I don't think you can by heart and write them in the exam. Just know that they show both polygenism and also show polymorphism or polyallelic nature. And at least two loci, like 1 and 2. Third one is also identified. Several genes under each of them. And under them, multiple alleles. Look at them. 28 alleles for this, 136 alleles for this. So together, so many possible combinations. 28 into 136 in a way. And here DQ 35 alleles, 106. So 3 and 8, 14. For locus 1, we have for B, 1431 alleles are identified. One location, there can be 1431 possibilities in one location. Like that, few tens to hundreds of alleles are present for each of these locations, each of these genes. And the final product is what? Final product is combinations of all of them. That is, it's complex. Combinations of all of them. Combinations coming from region 2, region 1, region 3. That is what would result in multiple possible HLAs. At least these are the effectively 6 categories here, 22 categories here, and imagine how many such categories are possible. Highly, highly polymorphic, highly diverse. Okay, right. So far, as I was telling you, I'm just trying to give you a picture about how diverse is HLA or MHC is. And where is this diversity coming from? Multiple locations, each location multiple genes, for each gene multiple alleles. If you want to write like a flow chart like this, multiple locations, 1, 2, 3. Each location multiple genes. We talked about A, B, C, D, P, D, Q, D, R. Multiple subregions indeed. Multiple locations, sublocations, and each region we have multiple genes. Okay, like that you see the diversity. For each gene, multiple alleles. Specific location, sublocation, multiple genes, multiple alleles for each gene. So enormous amount of diversity is created like that. There are some ideas why so much of diversity, but this is the an idea and a glimpse of diversity that MHC molecules show. Okay, right. Can we move on? Okay, don't try to remember. I don't think even I don't remember those numbers. I only know the diversity extent. It will be too much to remember for the exam. But just know the reason behind the diversity in MHC. Okay, what could be the reason behind this polymorphism? Why? What causes this? Maybe selection pressure. Like first of all, MHC molecules play an important role in immune system function. Maybe diversity is needed for tackling various types of pathogens. So given the types of variety of pathogens, maybe we need variety of MHC as well in our body. Varieties of pathogens. And also selection pressure that we may experience as host on pathogens, they diversify. Also in response to them, we diversify. In order to survive, they diversify. In order to us to survive, we also have to tackle the complexity of the pathogens. And what is the benefit of having multiple combinations of them? Heterozygote advantage. Maybe having two types of them. Let's say you have A type and B type, A1, A2, for example. So your body can recognize these varieties of molecules or antigens. You are at a higher advantage if you have two types of, or you are heterozygote. You have more diversity, right? If you're homozygote, what happens? You have A1, A1 you have. Otherwise, you have A1, A2 for heterozygotes. That gives us more diversity. And different MHC molecules bind different peptides. They can bind to different peptide molecules or protein molecules in the body and can provide us more broader immune responses. More broader immune responses are possible with this particular type of our heterozygotes have their advantage. And scientists have noticed that, so people who actually show delay in AIDS, they seem to have heterozygosity for certain MHC molecules. So people who are showing higher amount of MHC heterozygosity, meaning two different types of alleles from different parents that they have received, they tend to delay the AIDS progress. So meaning their body is able to tackle it better. Their body is able to tackle it better. Okay, this could be one reason. So maybe they provide us some advantage. The variety offers us more resistance to infections. And then even for host pathogen evolution, because pathogens are evolving and we are also evolving, right? We both are competing with each other in evolution in a way. So if we have a certain specific types of MHC molecules, what happens? Pathogens can easily adapt, correct? They can gain resistance easily to them. So people who actually have rare alleles or heterozygotes have an advantage over the people who keep having common alleles, because pathogens are adapting to this common alleles. So we need more rare alleles. We need more combinations to which pathogens cannot adapt. So the complexity of MHC may be defined by the infectious agents and diversity of the infectious agents. Are you able to follow this? The connection between MHC and pathogens. We're talking about why our immune system, those defining factors in immune system are diversifying, because pathogens are complex and more diverse. So to address that, to handle them, we are actually having that high amount of polymorphism with respect to MHC. And of course, it is causing us problem in accepting someone's organ, right? But I don't think MHC is designed to allow the organs. I don't think this organ donation or organ transplantation concept is not part of nature. This we as humans found out. So this MHC is not meant for that. MHC is causing a problem because it is so diverse. So not any one of us usually have same combinations, same match. It is very, very difficult to find a good match for that reason. Okay, so this is briefly about the MHC polymorphism, and I was talking to you about different regions or locations of it, and multiple alleles for each of them. I've given a brief overview on this, how polymorphic it is. And next I talk to you why polymorphism, why we have so much of polymorphism, briefly to tackle the enormous complexity that we have with respect to the pathogens. Next, use of it. What is the use of HLA markers? In what sense they can act like good markers? Understanding the immune system function that I talked to you about. Next, I'm going to also briefly put idea on the thing. Let me first do that. Graft organ transplantation, how HLA is used in organ transplantation. I kind of talked about this concept already. So we talked about transplantation of foreign tissues. So when someone's organ is taken into your body, your immune system can recognize it as foreign. So both the antibody mediated, which is known as humoral immune system, fluid based, or B cell mediated immunity, and cellular mediated immunity, like T cell mediated immunity. Both responses are normally seen in rejecting the graft. In rejecting the graft, both B cell mediated and T cell mediated responses are seen. Okay, so I was also telling you that MHC molecules are present, especially MHC1 is present almost on all nucleated cells or nucleus containing cells. So they are the ones to recognize whether it belongs to you or not. So immune system will check for whether this is my MHC or not. If it doesn't match, it knows that the foreign part has entered into it. It doesn't know why it entered. You cannot talk to it, right? So it knows that some foreign part came into it. So it said it is not, doesn't belong to me. Let me eject it. Let me target it by producing both antibodies as well as the cytokines. So cell mediated and humoral antibody mediated responses kick in, right? So that is why what do doctors do? Doctors would do proper typing. This is called HLA typing. What do doctors do? They would actually perform HLA typing, TYPING, HLA typing, meaning they are matching. They are finding how much match is present between donor and recipient. How much match is present between donor and recipient is what they normally find out. So is it possible to absolutely have the same HLA between two people? What do you think? Is it possible to have same HLA types between two people? How likely is that? Sunil?
It is very rare.
Very rare. Very good. Why do you think? Shweta, do you agree with him?
Yes, sir.
Why do you think so? Why can't two people have same HLA? Yes, do you want to answer this question? Anyone else?
Because of the diversity.
Even the diversity. There's so many different alleles that are possible, so many locations. The outcome is the complex combinations of so many locations, their genes and their alleles. It is very unlikely that we may share the same alleles with others. Even parents and children may not share it. Brothers and sisters may not share it. Okay, so it is impossible to have the same HLA typing. Then how do doctors manage to allow the grafts being transplanted? Fortunately, we don't need not all of them are important for it. I was happening to talk to one of my cousin who is a doctor and doing research on kidney transplantations at NIMS in Hyderabad. So she was explaining me one day that they don't match for every region. So there are a few regions seems to be more important over the other regions. So they have a priority of what regions to be matched. But in either case, even if you have prioritized the regions, okay, even then you will not have proper combinations that are possible. Then what do? As I was telling you, there is more possibility of having similar alleles if you are taking an organ from parent, one of the parent or one of the brothers or sisters. There's a higher chance of matching. If you're taking from someone else, like cadavers, for example, someone who died recently, their organ is preserved. In those cases, it's very unlikely. Correct? But blood groups are used to match it, first of all. Blood group has to match. Otherwise, it will add to more complications. Second, so what do? If there is some amount of a decent match had happened, they would give them immunosuppressants. These people who are receiving the organs, they are put on immunosuppressant drugs. Drugs that suppress immune system function. They are placed on the immunosuppressants. They say that even before few days of organ transplantation, they do it. And then I remember one of my friend went through organ transplantation for kidneys, I think 16, 17 years ago. He's on immunosuppressants since then. Put them at higher risk for infections and diseases, but they have no go, right? If they have to survive, if the kidneys are failed, what do? They have to use proper match. His mother donated the kidney to him. And his mother is also doing all right, and he's also all right. His mother donated the kidney, okay, and he still is on immunosuppressants. He's more and more prone to infections. He has to be really careful throughout his life. So like that, they're managing with some amount of matching plus immunosuppressant drugs. Okay. So class 1, class 2 are most important from the SLA type. Immunosuppression treatments are normally used to suppress that immune system responses which can reject the organ. Okay. So this is briefly about if they ask you talk about HLA typing role in organ transplantation. This is the most important aspect of HLA or MHC. So I wanted to give you a brief idea on this. How does that happen? So in case they ask you to focus on this aspect of MHC, do talk about these points. Any questions on this? This is something that you could follow.
One question. So one who receives it, the person who receives it has some immune response. The person who loses it, does he have any again a B cells T cell response? Because something has been missed out.
When does immune system matter here? Whenever a foreign part is coming into the body, only then your immune system tries to identify whether it is foreign or belongs to it, and it tries to attack it. So here we are focusing on what happens in the recipient, not on the donor side. Because donor side, even loss of something doesn't matter because there is no immune system components or new antigens are introduced into their body. Correct? As such, MHC would help us identifying the foreign particles and destroy them. But the problem is more severe when we are talking about transplanting things. So here, more than what happens on the donor side, what happens on the recipient side is the thing that we are worried about. In the donor side, there's nothing much that is going to happen because his body part, let's say someone's kidney is taken, donor's kidney is taken. MHC won't recognize it anyway differently, right? That doesn't change. Its way of recognizing its own tissues doesn't change. But when someone receives it, their immune system make the problem here. That is why we want to have enough match, and absolute match is not possible. So we use immunosuppressant drugs to make his body to get adjusted to the new tissue. Right, Sunil? Did I answer your question right, or were you asking anything else?
Sir, one more thing. So, if there is any disease, let's say cancer, so again we have the same response like we talked in the session. So we again have the role of HLA.
Right. Yes, anything happens in the body, yes, cancer happens to our own cells, right? You don't see too much of a difference in MHCs there. If a viral particle came, bacterial infection came, protozoan came, then they can recognize them as different from us. Okay, so in those cases, what response TL per cell response, TC cell response, B cell response is defined by them. Okay, but cancer cells may not cause any difference to that because there are own cells which lost the control on cell division process. Okay, right. So yes, this is briefly about the organ transplantation part of the HLA. So we'll talk about now distribution of HLA alleles, what is known about their distribution of alleles and what are the other applications of them. Okay, so distribution of the alleles, one study from 2008 that has shown that, so there are a few differences that you may see in different parts of the world. Okay, so every region there may be some amount of specific types of categories of MHCs are found specific to particular region. So region wide HLA alleles seem to be noticed, which is no surprise to us. Few regions, these alleles seems to be more common because they tend to marry, they tend to share the gene pool. They tend to have same alleles being distributed around, correct? So large numbers of regionally restricted alleles exist. So region specific alleles are there. Some alleles are more common to Europeans, some are more common to Africans, Americans like that. They have noticed. Only a few of the HLA-B alleles, HLA-B, that they name them differently. 40 is to 02R, 51 is to 01G. They have, because given the complexity, they have to name them differently. Okay, so only if you have the HLA-B, A, B, C we talked about, only few HLA-B alleles are shared across regions, meaning HLA-B is seen in many regions around the world. So some HLA-B alleles are found commonly in many regions, but otherwise regional specific alleles are found. HLA-B alleles are shared across few regions, but only few of the HLA-B alleles. So HLA-B is somewhat shared, whereas HLA-A, C, and D, some of the D of course we have a lot of variety. They say D or B, but in contrast to B, A, C, and part of the D alleles are more common. They are more commonly shared around the world. So some amount of distribution is same, some amount of sharing. Some studies have indicated that these alleles are more common in this region, these alleles are more commonly found throughout the world or many regions in the world. Such information is available. Okay, so just briefly remember that there's so much of diversity. What causes the HLA diversity? Then you know that the distribution of the HLA alleles also varies a bit. Few alleles are shared throughout the world. Few alleles are less common in the world. HLA A, C, TRB1 more common, B is less common, and other varieties are also less common. Okay, they are most commonly found in few specific regions.
Sir, if there is a medical tourism, so if someone comes to India to get the kidney, so that would be very different.
It will make it really difficult. Exactly, you're absolutely right. So matching between them will be very, very challenging. So someone coming from America to get the transplantation done in India, finding a donor here will be very, very challenging. So these are the technical, analytical aspects that you have to think. That's a very good point. Okay, so what type of implications we may have of knowing HLA type? Is it really useful to us in planning such type of transplantation? Very much. The diversity, polymorphism in a given population is very important because they can define the success rate of transplantations. Okay, right. So any applications, any specific applications of HLA? We already talked about some of them. Okay, anything that we have missed? So immune responses to pathogens. So how our immune system functions is defined by the HLA. Are you done writing what I was talking to you? Or should I wait? Excuse me. Right. So applications wise, immune system responses. Already talked about MHC 1, 2 categories define how immune system functions. So next, immunogenetic systems is highly informative for anthropology. We all kind of already touched upon those aspects. How is that useful in anthropology? So patterns of HLA genetic variation. So variation can give us clues of how people are distributed, what are their origins, who are more related to the others, like migratory studies. Those components can also be studied using HLA. Given the diversity, if there is more similarity, meaning they're more related to each other. Maybe there are similar ancestors that they have. So patterns of this in HLA is very useful tool for us to understand spatial and demographic human population, demographic distribution of human populations. Where do they live, where they might have come from, and why do they have same correlation with respect to HLA is very, very important. So HLA can be a great tool in studying human populations in anthropology. Given the diversity, it can give us more clues about human populations and their distribution. Okay, I already talked to you about organ transplantation. So immune system responses, organ transplantation, anthropological use in in studying populations. Immunogenetics also we talked about them, right? Immunogenetic system and medico-legal problems. We also I briefly mentioned with respect to blood groups. So blood groups by alone cannot solve the problem, but together with HL is a good source for paternity disputes, because not like if you picked up a biological father and a child, so child should have more similarity to the particular biological father. HLA cannot mislead you. Given the diversity, you cannot find that variety unless that the genes are received from that parent. So HLA can also be a very good source for solving paternity disputes. So HLA, blood groups, even better than blood groups. But blood groups can make it easy for us to solve the problem, but they cannot completely solve the issue, but HLA can help us. So paternity disputes can be solved using HLA typing, along with the other applications I mentioned to you. Okay. As a value addition to this, I would like to add some information I picked up from a news article of HLA connection as a marker to certain diseases. So in diseases, can we use HLA? So I found association of HLA with certain diseases. So category of HLA and the disease that is seen, like type 1 diabetes. Specific varieties of HLA alleles are more common with people suffering from type 1 diabetes. So D versions, DR3, R4, DQ8, so DRQ, we're talking about it, PQR. So some of these combinations are more commonly found in type 1 diabetes people. And interestingly, most of the diseases we are seeing here are autoimmune diseases. They're autoimmune because HLA is responsible for maintaining the immune system, right? There is a key role in playing with the immune system. So immune system losses, like a type 1 diabetes and autoimmune problem. Graves disease, I'm not sure if you heard about. Some people actually have hyperthyroidism. So thyroid problems can be because of autoimmune as well. Graves is one thyroid issue. This abnormal thyroid gland. So in Graves, they produce more amounts of thyroxine hormone, excess thyroxine. So that is called hyperthyroidism, right? So Graves simply is hyperthyroidism, one condition of hyperthyroidism, Graves disease. And people with Graves tend to have DR3 type of HLA. So like that, scientists have found with respect to different autoimmune disease, different version of HLA allele that is maybe responsible. So some correlation between HLA allele and the disease. So can we use such an HLA allele as a diagnostic tool to see if they develop this problem? Because Graves and these things are not present by birth. They only show up at later stages of life. There's some trigger sometimes. But these alleles can help us identify beforehand if this person can have a problem like this. So allelic correlation with the disease.
Sir, are you saying that HLA allele are born before the disease itself?
HLA allele is there. As soon as we are born, our HLA combination is defined, right? We have certain genes. As soon as we are born, we have that genes defined. So, but the problem may show up later. So Graves disease may show up in late 20s or 30s. But you can identify if the person has DR3. For some reason you suspected it. Look for DR3 allele. DR3 allele may tell you that this person is more prone to have Graves disease. So what is the benefit of it? You can actually handle, because thyroid problem is very much related to how you handle the food, what type of foods you eat. You can deal with the food by dealing with the food, by choosing the food. There's some compounds called goitrogens, which actually suppress the activity of thyroid gland. That can be handled by knowing the particular type of HLA allele. So HLA can help us, okay, in detecting the particular problem well beforehand. So could be used in diagnosis of diseases, especially in autoimmune diseases because of its role in immune system function. Okay.
Autoimmune means
Our own immune system thinks or detects our tissues as our own foreign tissues. So our immune system attacking our own tissues. So type 1 diabetes, pancreas is attacked by our immune system. So pancreatic cells die, insulin production decreases. So our immune system attacking us. It's supposed to attack the foreign particles, right? But sometimes it attacks our own cells. That is where these problems happen. Okay? Right. So these are the various applications of HLA alleles. Okay. Need that. So this is briefly about HLA or MHC. So what are these MHC? What is their role in immune system functioning? And where is the genetic diversity is coming from? And the brief idea about the diversity and the reason behind diversity. So applications and distributions of HLAs we talked about. Right, I have not seen any specific area of question, but if at all they ask, I think most importantly, why is it so polymorphic, or pick up a very highly polymorphic region in humans, discuss about their use and their distribution, talk about it. Otherwise, I covered all the major subheadings. What is HLA? Where is this information present? How are they distributed? What are the causes for polymorphism and applications? So organ transplantation may be one very important application of this. Okay, right. So this is about MHC or HLA. The next one, briefly, I would like to talk to you, and that is the last topic for today, is the haptoglobin. If you see in the syllabus, they have not specified it very much here. They say HP after HLA, so we are going the same order, ABO, RH blood groups, HLA we finished. HP we are talking about. What is HP? HP is haptoglobin. Haptoglobin. So what do you need to do for now on? Write a brief summary of what I'm explaining. This should be it. You don't have to refer to anything else. Okay, I'll tell you what is HP, how polymorphic it is, what is the use of it. That's it. Okay, so what is that protein we're talking about? How much polymorphism does it show? Nothing shows as much as polymorphism as HLA. We're done with the most diverse one. Okay, HP. What does HP stand for? HP stands for haptoglobin. HP in the syllabus stands for haptoglobin. Okay, so what is haptoglobin? Haptoglobin is a protein found in our blood, in the plasma, in the blood plasma. So it's a plasma protein. Haptoglobin is found in our blood, mainly in the plasma. Blood, we have a fluid part and the plasma part. Plasma is the fluid part where we find proteins. Otherwise, cells, RBCs, WBCs, platelets will be there, plus fluid will be there. Fluid contains also proteins, sugar, proteins are there usually. So it's a glycoprotein, meaning protein attached to a sugar attached to a protein, mainly protein to which sugar is attached. Okay, don't worry if you don't have to remember the molecular weight, but 90,000 units say. So what is it doing? Where is it produced? So this HP protein is produced in the liver. It is synthesized in the liver. Hepatocytes means the liver cells. It is synthesized by the liver cells. As you see, haptoglobin is synthesized in the liver. So it looks very much like how hemoglobin is. Hemoglobin is also contains 2 alpha chains, 1 beta chain. Okay, the image is slightly wrong here. So this is how normally hemoglobin looks like. Hemoglobin looks like 2 alphas and 2 betas. But here it has 2 alpha chains and 1 beta chain. Okay, so mainly found in the liver, produced in the liver. What does it do? It is an acute phase protein. I'll tell you, I'll explain you what does that mean. Okay, let's see what is it doing. What is the function of haptoglobin? So its main role is to bind free hemoglobin. Remember, RBC also contains hemoglobin. Haptoglobin is an addition to hemoglobin. Whenever hemoglobin is released, haptoglobin will bind to this. Haptoglobin will bind to the free hemoglobin, which is not bound to the oxygen. Why is that? So otherwise, hemoglobin can get through the kidneys. It can go through the part of the kidney called glomerulus. So it's a region where, like we have in the kidneys, several tiny filters. Within the kidneys, we have several lakhs of filters called nephrons. You might have studied them in high school, nephrons, right? So within the nephrons, we have a portion called glomerulus. So hemoglobin can easily move through this glomerulus part or filters of the kidney in a way. And when it passes through that, it tends to settle in the kidneys, causing kidney damage. So if you let the hemoglobin pass like that, it can settle in the tubes or tubules, tiny tubes connected to the kidneys and causing kidney damage. So we don't want hemoglobin to pass through it. Looks like haptoglobin job is to capture the hemoglobin, capture the free hemoglobin. So what happens when HP bound to hemoglobin, it will become too big to move through the kidneys. Kidneys glomerulus part. So filters, it cannot fit to the filter. It will be filtered out. So it will remain in the blood. So haptoglobin will bind to hemoglobin and will not let it go through the kidneys to the nephrons, thereby preventing the damage that it can cause to the kidney part. One second, let me bring another slide for this. Okay, so with respect to this, we so far studied that it's a protein that is found in the serum, the plasma, blood plasma, made in the liver. So mainly its job is to bind to hemoglobin, correct? So it binds to hemoglobin and thereby preventing the hemoglobin going to the kidneys. Okay, so renal damage and kidney damage is protected here. So that is why one thing scientists notice that, so the free haptoglobin, because haptoglobin job is to bind whenever hemoglobin is present, right? So when hemolysis happens, our RBC lysis take place, more and more hemoglobin gets released, right? So haptoglobin will bind to the hemoglobin. So what happens to the free levels of haptoglobin in the plasma? It will be very low. So meaning that haptoglobin bound to hemoglobin, something is happening inside the body. Maybe free hemoglobin levels are increasing. Why? One reason could be damage to RBCs. So plasma levels of haptoglobin can be used in detecting or monitoring that scenario. Okay. So plasma levels of the haptoglobin can be a good tool for detecting the condition with respect to hemoglobin and the causes behind it. Maybe lysis of RBCs can be detected using it. Okay, right. So the next point, I was telling you briefly, acute phase protein. It is an acute phase protein, meaning whenever our inflammation response kicks in, inside our body some damages happen, reddening, swelling of the various parts are happening, which is called inflammatory response. In inflammatory response, you will see increase in haptoglobin. So it's an acute phase protein, meaning in acute conditions like inflammation, so haptoglobin levels increases. That is a sign that we have inflammation is happening in the body. And decrease in acute phase protein like this is a sign that hemoglobin, something is wrong with the hemoglobin production. Okay, like that, haptoglobin is of some clinical use to us. So clinically, this can be used in monitoring what is happening in the body.
Sir, is it platelet?
Platelet, what? This.
Acute phase protein, is it platelet?
No. Platelets are different. Acute phase proteins are this group of proteins that our body makes during inflammation process. Platelets are different cells. Platelets are meant for clotting. They are cells indeed. These are proteins. So platelets are cells found in the blood, mainly meant for blood clotting to take place. But these are proteins. They are produced during the inflammatory response.
Sir, they are also sometimes used to assess the inflammation.
They are sometimes go to.
Assess the inflammation, platelets.
So even platelet count increases with the
Yeah.
inflammation. Yeah, that is possible because inflammation, many events take place. Okay. So haptoglobin is one such protein which does it, but it is not a platelet. Platelet is a cell. Okay, because inflammation may lead to some damage and the bleeding may happen. So maybe platelet count goes up because of that. Okay, but acute phase protein is different from that. Okay, like polymorphism. Let's look into the polymorphic nature of haptoglobin. How polymorphic it is, some genetic information on this. So haptoglobin is present on chromosome 16. It is present on chromosome 16, HLA on chromosome 6, haptoglobin on chromosome 16. Okay, but two types of mRNAs are formed from this gene. So the gene is present on the chromosome 16. One mRNA produces two chains, alpha and beta chains. As I was talking about alpha and beta chains. So alpha and beta together in combinations gives rise to haptoglobin. Two alleles. This is very less complex compared to HLA. Only two alleles are present here, HP1 allele and HP2 alleles. So haptoglobin, HP1, HP2 alleles. Two alleles are present. They're co-dominant in nature. What does it mean by co-dominant? Like AB blood groups are co-dominant. Both A, B expresses. So two alleles, three distinct genotypes. Two alleles, three distinct genotypes. What are they? HP1. So one allele 1 homozygous condition, 1 and 1. Allele 2 homozygous condition or heterozygous condition. Correct? So two alleles HP1, HP2. So what are the possible genotypes? Homozygous for HP1, homozygous for HP2, and heterozygous for HP1 and 2. Three possibilities. So I don't think you need to technically know this, but gel electrophoresis technique, do you remember? We talked about how they can separate DNA. They can also separate proteins and alleles. So they're showing here the difference between 1.11 to 21. So homozygous condition, heterozygous condition, their pattern will be very different. How alpha 2 is seen in these two, and whereas in one, you don't notice the alpha 2. alpha 2 protein, alpha 2 polypeptide. Okay, so 2 alleles, 3 possible genotypes and 3 possible phenotypes out. Okay, so what about the distribution? So for each of these markers, as I was telling you, what is it doing in the body, how polymorphic it is, and what is its distribution. So seems like races specific HP1, HP2 alleles are there. Some HP1 alleles are more common in some races, some alleles are more common in other races. So where HP1 allele is increases from Southeast Asia to Europe and Africa, and from Asia to America. So the distribution of HP1 allele seems to increase from southeast part of the Asia to the Europe and Africa, and from Asia to America. So HP1 allele is more common in those regions, in the European regions and African regions and American regions than Asia. Okay, and especially in the Americas, South America, within South America, there is highest frequency of HP1 allele. They have 70%, 0.7. You see 0.7 probability, or 70% of this ratios that you see in the South American population. So they have mostly HP1 alleles are more common. But as we were seeing, HP1 goes from the Southeast Asia to the Europe and America, right? So Asian populations have low amounts of HP1 because it goes up from Asia to Europe and Americas. So only 0.2%, even in India, we have less of HP1 alleles. So in a way, intermediate in the European populations. European people have about 40% of HP1 and about 60% of the HP2. As per a study published in 2007, I gathered this data because no major sources or textbooks talk about this information. Pinath roughly gives some information with no logic. So that is why I was telling you this is the only resource that you can probably easily access. I have original papers on this if you are interested, but given that they're not asking too much on this in the exam, just the summary that I'm explaining in the class is more than enough for you. We talk about how they are distributed, HP1, HP2 alleles. So how are they changing from one region to the other region I talked about. And HP1 is less common in India, more common in other countries, especially South America. So this might be helping us to know or asking a question, is it originated in India? Some studies are saying that maybe 2 million years ago, this HP2 allele might have originated in India. There may be some advantage of keeping it which is not known, but HP2 is definitely the dominant version in India. Okay. So what are the uses and applications of this? So variation with age seems to be happening with it. Okay, because one aspect they're asking for them, how they vary as a genetic marker. So with age, haptoglobin seem to, change. So it begins at the 3 months time. So haptoglobin is not found before 3 months. This expression only begins after about 3 months in the blood. So it begins to show its presence or expression starting from 3 months of our life, and then it slowly increases up to the age of 20 years. So about 300 to 200 milligrams per decaliter or 10 ml are normally found. So age variation may be seen. So this can be a useful tool. Age can be identified using haptoglobin levels. It won't be that accurate, but a correlation can be seen here. So age-based variation can definitely be seen, which is a useful tool with respect to haptoglobin, along with the detection of inflammatory response in the body and hemoglobin, free hemoglobin levels in the body. Okay. So any other uses, applications of this? I was telling you already. So people are suffering from hemolytic anemia, RBC lysis. By measuring haptoglobin, one can identify that there must be something to do with the RBC lysis. So free hemoglobin content is there. So haptoglobin levels have decreased. So haptoglobin levels can help us, as I already told you. And inflammatory response. So presence of haptoglobin levels may also indicate some problems like heart attacks, rheumatoid disease. So inflammation in the joints, rheumatoid disease it is called. Inflammation in the joints. Heart attacks, rheumatoid disease or problem autoimmune disease related to the joints. And diabetic nephropathy, meaning sugar-induced nervous system damage. So in those cases also haptoglobin seems to increase.
Nephropathy is kidney.
Sorry, yeah, nephropathy. Damage to the, did I say nervous system? So, sorry, sugar, diabetes induced kidney damage. Because diabetes can be dangerous to kidneys as well as heart. So when people with high amounts of diabetes in COVID time also we are hearing it, right? People who have kidney damage and higher amounts of BP, they're higher risk because diabetes tends to affect the functioning of these parts of our high sugar levels are toxic to us, especially to the heart as well as the kidneys. So diabetic induced nephropathy and kidney damage, rheumatoid disease, so joints, joint pains, especially through autoimmune diseases, heart attacks. In these cases, haptoglobin level seems to be high. Okay, and I already told you inflammatory response, autoimmunity responses. So in these cases also, haptoglobin levels increases. So population level analysis, disease conditions, specifically inflammatory conditions, they can be studied using haptoglobin levels. Okay, right. So this is where I want to stop here, the class. The next class, I'm going to talk to you about transferrin. So if you want, you can make a note of these things, but I think these are a bit more, this just the idea about what have been found out by the particular alleles in their immune responses. So it's up to you. If you want to make a note of a couple of them, I'll mention it here. Okay, in various studies that they have shown that specific allele is connected to the specific disease outcome. HP2 phenotype, HP2 to homozygous HP2 condition, higher mortality rate from HIV. So people are more prone to HIV if they have HP22 condition. Previously, we talked about a condition where disease progression in HIV is slow. Similarly, some may promote it. HP22 combination seems to increase the death rate from HIV, as per a study. Whereas HP11 makes us more susceptible to malaria caused by falciparum, severe form of malaria. A study by in 1998. HP11 is more common in falciparum malaria. So people who are susceptible to that tends to have HP11, homozygous condition for HP1. Okay, so this is value addition in case if you want to write more on this. So 2 talks about the mortality from HIV, 1 talks about the problem with malarial falciparum, and high amounts of HIV is also seen in cancers, lung cancer, breast cancer, etc. Okay, all right. So we'll stop the class here. So tomorrow's class, I'm going to wind up the rest of the markers like transferrin, gamma globulin marker. We are going to talk about blood enzymes. Few examples of blood enzymes I'm going to talk about. Okay, with that first part of the 9.6 will be done. Then we'll move on to the physiological characteristics, hemoglobin levels, body fat, pulse rate, respiratory functions. How do they change in us, and gender to gender differences if there are any. Okay, we're going to focus on that. Right. So as we discussed at the beginning of the day today, so we're going to have the test on Sunday. So test is moved on to Sunday. So we'll finish 9.6 this week. So mostly I'm going to start with ecological anthropology 9.7 this week. Towards the end of the week, we are going to begin with 9.7 this week. Okay. Right. Any questions, guys, from today's class?
Sir, Venkat sir mentioned that during the upcoming current affairs program, which is about to start on Feb 6th, mentioned that you will be taking classes during the morning because he's planning to take it in the evening.
Yes, sir briefly mentioned that few days he might wants to switch the time slots. So where he wants me to take the mornings, but he has not told me exactly when. But he said he might need some evening classes. Would that disturb your any schedules? You're also part of that class? Some of you maybe?
Doesn't disturb any schedules. Just I wanted to know.
Yes. Sir mentioned to me that he might take some classes in my slots in the evening. Yes. Okay, but I think you will be informed about that. When is that? Sir, whenever sir tells me, I'm going to announce it on the group. So when is my class going to be in the morning? Okay, if you guys have any problem, we'll discuss that and see what this can be done. But of course, you have access to the videos. Only part you will miss is the discussion part, right? So in case if something like that happens, we can discuss it separately. Good for today. So right, guys, good night. So we're going to do class tomorrow at 4 o'clock. So bye now.